and participate actively to the workshop, just shoot me an email during the session before 2 p.m. and I will send you the credential to be able to join the workshop. We want this workshop to be successful, and we need your active participation. So please shoot me an email, and you are more than welcome in this afternoon workshop. Now we'll move to the second one, and the second one made some kind of nice performance. She is Rosario Sanchez-Martin from Reggio in Spain. Hi again, Rosario. And you made the performance of having 2 submitted abstracts and 2 selected abstracts.
Yeah, I can see that, yeah.
And very nicely ranked among all the applicants. So congrats for that. And so you have some kind of double talk. And if you don't mind, I will now broadcast your lecture. And you will be here for the Q&A. Thank you again for this.
Okay, thank you.
Hello everyone, and thanks to the ATPN for this opportunity to share our results in this Pitch Me At session and be able to present you these 2 projects of nano device that we are developing right now for nano diagnostic applications. First, please let me introduce ourselves. We are NanoChem Biolab. We are working in the Faisal University of Granada, Junta de Andalucía Center for Genomic and Ecological Research, GENYO. And we are located, this is our team. This is the member of the team that we are working every day. And here is the place where we work. We are in the PTS Granada, that is the Health Science Technological Park. And in here we have our research center, but we are lucky enough to have in the same park, in the same place, we have the hospital. And what is even more important for us, we have the biobank that is the one that is giving us the samples from patients from the hospital. So we have the way to have the samples in a safe way. And then we have also biotech companies, small and medium enterprise companies in a building, also here in the PTS. And here are companies that we work with them, such as Nanogetic and Destina. And also the animal facilities, between other things. So now that you know a bit about us, I would like to present these 2 ideas, these 2 projects that we are working. So the first one is related to polymeric nanoparticles for diagnostic applications and how this works. The mission for this is like we're looking for to have a rapid and accurate cancer diagnosis and prognosis together with monitoring cancer treatment efficiency using liquid biopsy, using blood samples from patients instead of using the tumor tissue. So the mission for this is we are designing and implementing an innovative nano device that would allow us to isolate, label, and analyze the individual exosomes from these samples. Okay, so what are the medical issues we are trying to address with this nano device? The thing is, it has been described that inside the exosomes that they are coming from tumors, and they are in these blood samples, there are proteins and microRNA levels that are very related to the tumor stage. So it could be a fantastic tool for early diagnosis and specific intervention. So, but the thing is, people are working with nanotechnology and apply to exosomes or any kind of a method to isolate exosomes, you need some challenge, 2 of them. So the first one is like you need to isolate them from the complex biological mixtures. And then the second one is when you have isolated them, you need to handle them. And this handling is difficult because if you need to do a multi-step process to analyze them, then it's complex to carry out. So what is the current solution? You have exosomes isolation methods currently available. I will say here you have all of them. I will say people use ultracentrifugation, but also do differential surrogate precipitation between other affinity methods using magnetic beads, chromatography, etc. The thing is they have advantages and disadvantages, everything, but I will say that one of the main concerns are time-consuming, that there are some of them, like ultracentrifugation. Also, other options can be also only used for small volumes, and there is still a gap to find a good way, an easy way to isolate these exosomes. So here is where we come. Our solution will be to protect, to handle exosomes. So this project will be to handle the exosomes using this nano device to isolate them and enable multi-step learning of both the proteins and microRNA. So how does this work? We are going to have this nano device. We are going to have a tracking part. So the tracking part could be a fluorophore or a metal tag. So depending if it's a fluorophore or metal, we'll have different applications. And then we have the exosome isolation by antibody recognition. So why our system is, this solution is unique? So the idea is because this method is very versatile. So the protocol that we have developed so far, we can use it for conjugated antibodies specific to different kind of exosomes, and the protocol has been already optimized and validated, and we can use different antibodies. So it's a versatile platform. It's easy to handle. You just need to have a centrifuge. No ultracentrifugation is needed. So you just need to have a centrifuge in your lab. And also the cost is low cost because you don't have very complex system to work with. And the more important thing is heartless. Is mean the exosome after isolated, it keep intact. So it's keep it harmless. So it's so you can carry on and doing all your analysis, having a nice sample of exosomes. So saying that, the evidence that we have so far is like we know we have prepared them, we know how to prepare them, we have the protocol is optimized already, this has been patented, so the submitted has been patented already. We have the efficiency of conjugation, that we have checked that the nanoparticles, the antibody is properly conjugated to the nanoparticles, so by different ways. So if anyone's interested, I can describe later on. Then we also have used different techniques to check that this is working properly, this nano device to catch this exosome. And here are the results. And the thing is, we are working with 3 different teams in Spain, in Malaga, A Coruña, and Granada. So each one with the Dr. Eduardo Garcia, Dr. Mayan, and Dr. Marchal. And each one of them are working with exosomes of different nature and studying different disease. So they are using different antibodies to be conjugated to the nanoparticles. So we are having different approaches. Then the next step, what we do, so we will scale up the nano device. This is something that we are working on this. We are trying to evaluate the versatility of this nano device because there has been described, you have here this paper where there has been described many biomarkers that there are different biomarkers that you have in the exosomes when they are coming from different tumors. So we're working with that. We have also another project where we are evaluating this device to use combine for diagnosis of nucleic acid detection, and we have recently received funding for that. I won't go deeply through this, but the thing is, we will be able to take these exosomes and then see what is going on about microRNA levels and protein levels in these exosomes coming from blood samples of patients. And the last thing that we are looking for to do next step is to do a multi-center validation. So for this is what we need the CTPN and people that are interested to try to use this exosome, these nanoparticles to isolate exosomes. So we are more than happy to work with them, because the thing is to validate what you are working. You need to use to do a multi-center validation to check that the protocol is working okay. So I don't go more on this. The second project that I wanted to share with you was Nanobarcoding reagents for tracking biomass cytometry. So what is this one about? The mission of this project that we're working is to personalize treatment, to be able to stratify patients. Why we need to stratify patients? You have the population, but you can see that it has been described in many places that you have patients that they are not with the same pathology. There are patients that they will respond in a different way to the same treatment. So you need to stratify the patient to decide the right treatment. So what is our mission? Our mission is we are designing and implementing a nanobarcoding tool to allow multiparametric mass cytometry analysis of individual cells. So all these individual cells, they will combine and they will be analyzed in a multiparametric way. So the medical issue that we are trying to address, I just said, so very briefly, will be very positive to identify targeted intracellular pathways in the type of cancer that they are very high prevalence and high lethality, and this type of cancer usually they are resistant to current treatment. So it will be very nice to have this analysis and to know more about that. So, and then we can identify the most effective drug combination depending on this stratification of patients. So quickly, current solutions that is working for this multiparametric analysis of individual cells is mass cytometry. So why mass cytometry? Mass cytometry is really a fantastic method. The main thing is like, we work with flow cytometry. We are used to flow cytometry. The idea is you have tag, you have your reagents, your antibodies, any ingredient that you use, they are tagged. You have a label with fluorophore. So what is the difference here? The labeling is done with metal. So each metal will have an individual and unique peak in the spectra, in the mass spectrometry. So you will have any more compensation and overloading of signal. So you can combine many parameters at the same time. And this is fully working, okay? So there are even 60 markers available. So there have been many mass tag reagents that are developing to do this. So in this context of mass cytometry, that is allowing this individual cell multiparametric analysis, is where we are coming with our solution. We have designed and developed some metallofluorescent nanoparticles for live cell barcoding. So many of the reagents that can be used for nanoparticles, so they are for fixed cells. And anyway, so this was the idea of the project. So you have different cells, you label that, you put this nanobarcoding, so each type of cell will have a different barcode, then you put them, you have all of them together, and then you can do the staining with the specific metal tag antibodies that you want to interrogate, or you want to interrogate the microRNA in these cells, or anything that you want to interrogate, to all of them together, and then you will have your results, your multiparametric results. So why our system is unique? The main thing is like you have your cell, we do this barcoding with our nano device, but we don't need for this nanoparticle-based live cell barcoding. It does not rely on protein expression. So the physical barcoding is independent of receptor level of expression, so it's very uniform, and therefore it's not limited by the cell type, because independently of the receptor level of expression of each cell type, you will have the same labeling, so it's a universal cell barcoding. Then we also have seen that it's compatible with other live cell barcoding, because does not react with cellular proteins. It's versatile. We can use this for long-term tracking, because it's stay a long times in the cells. Not toxic, specific, and sensitive barcoding. So the level of evidence, we have done this experiment where we combine two cell lines, two breast cancer cell lines, one with one code, the other one with a different code. One code is palladium isotope 106, the other one is palladium isotope 110. However, the thing is you have two barcodings, then you combine the two cells in different proportions, and then you analyze them. And the thing is, we can see how we can keep this live cell barcoding is accurate and is efficient, and you have perfectly the proportion of the barcoding is keeping perfectly matched with the combination that you have done. So then we have done some experiment with Professor Carinola and group in Stanford University. This was a collaboration. One of our researchers was there. And the thing is, we have applied this for ovarian cancer cells and try to do this barcoding and do some profiling of different expression of different markers in the cell, and has been working quite well. And the thing is, next step, where to go? Again, scaling up, we're working on that. Evaluation of versatility of the nano-device. So we are a part of Palladium 106, 110, we are finding other isotopes. We are implementing this nanobarcoding to track treatment efficiency. This will be an application. And again, like I just told you 1 second ago, multi-center validation. We are looking for things that they have to interrogate something that they need to combine several cells and see how they all together respond to a treatment, and see how any kind of multi-parametric experiment, and we were very happy to share with them. So knowledge funding, many people, many projects, and many network, COSACTION, TPN, Nano2 clinics, Nanocatalysis in Spain, many research funding, the companies that we're working with, Nanogetic, and Destina, and that's it. So thank you very much. And I'm happy to answer any question or to keep any suggestion. I'm very happy to be here in this pitch me section. So thank you so much indeed for this. To take it.
Sorry, Rosario, I just cut it the end, but you were saying
it was too much by.
No, that was perfect. You were saying that you would be happy to take more questions. So let's see if we have questions. First of all, congratulations. That was excellent talk. Very nice presentation of two excellent technologies. So we have a question from Professor Alonso. And the question is, nice presentation by Rosario Sanchez. CyTOF looks like a great tool. I would like to follow you, to follow with the conversation with you. I thought it was a question, it was a comment.
Okay, brilliant. Yeah, it will be really nice. I mean, the expertise of Maria Jose Alonso is doing translation is really well known, and I'm really delighted to talk to her and to talk more about this CyTOF technology. And yeah, and I would like, this was my message from the video, from the talk. So it's a nice technology. It's not available in many places right now, so we are open to work with anything.
Fantastic. Is there any question on one of the two technologies right now? Otherwise, we are going to move to the next one.
I sent you one, Alex, because I can't write in Q&A.
Because you're a panelist.
Yeah, exactly.
You are too high in the hierarchy, you know that. So let me check. Oh, please ask it live, Ruth.
Yes, please.
Ruth?
Yeah.
Can you please?
I couldn't do anything with my mic. The question is that your next step is the scale up of the devices. And do you see any specific challenges, and have you planned to apply for one of the OETBs?
Will be nice. I think will be a good opportunity to apply. I have to say that in this case, we are a little bit more happy about that, because we don't need the GMP because it's for diagnostic. So we don't need anymore, because our previous project that we are working on it is for a nano device to be used for therapy. And in this case, the GMP make everything much more complicated and the sterile thing. So, but yeah, it will be really, really nice. I think we could try and apply. It will be really nice to have some, because the thing is, the big thing about all this stuff, scaling up. We are doing a small scale with Nanogetic, that is a company that, small biotech that I'm the founder and right now chief advisory board. But yeah, it will be really nice to have more advice, because I think this is one of the places where all people working in nanotechnology need help with the scaling up. So yeah, maybe I can ask for some advice later on and see how to apply.
Yeah.
Thank you, Ruth.
Well, it reminds me Malaga for the ETPN 2017 great event, where some of the projects that we've heard today were created like by very informal discussions in Malaga. And GENYO has been a great partner in Malaga to help us in the organization, even in the funding of this event. So thank you again for that. And I hope that this virtual event might, let's say, promote some new interactions for new projects, even if it's better when we meet in person indeed.
Okay.
Thank you again, and congrats, Rosario.
Thank you so much, Alexander.
Now we move to the next pitch, which is from CYBERNano, and Jeanne Deleforterie will give the pitch. Jeanne, are you here?
I'm here. Hi, everyone.
Hi. So you will speak about a project which is called a Dendrogram clustering of lipid nanoparticles. And I will just broadcast your lecture right now, and you will be available for questions, right?
Yeah, perfect. Thank you. Hello, everyone. I will present you an abstract we have submitted for the ETPN virtual conference named a Dendrogram clustering of lipid nanoparticle done by CYBERNano for the Pitch Me Up session. So in the last 2 decades, nanomedicine has grown into interest, especially for cancer therapy and or diagnostic, for instance. Scientists have been particularly attracted by the use of lipid-based nanoparticle as a new pharmaceutical delivery system to overcome biological barrier. Indeed, thanks to their size, component shape, or even their lipid structure, and they were shown to be more efficient and effective than pre-existing delivery systems. For instance, due to the fact that their main components are lipid, mostly well-tolerated component, this allows them to optimize the pharmacokinetics as well as the bioproduction when introduced into human organism. As a result, we have elaborated a new approach to classify different types of lipid nanoparticles according to their formulation and manufacturing features. So through this abstract, we have tried to reduce the confusion in terms of terminology and composition of all LNP through statistical analysis based on their design and manufacturing features. Indeed, a variety of LNP exists, as you can see here in this figure. Consequently, it may be difficult to differentiate them and notice their difference in the case of a non-expert in the field. So various articles in the literature are devoted to LNP, but only a few of them have tried to classify them. In fact, generally, they focus on the same type of LNP, such as solid lipid nanoparticles or liposomes, but a lot of other types existed and are not as well as emphasized, thus bringing more confusions. So here on this table, we can see that we have tried through the bibliographic research to classify all the type of LNP. In the row column, you can see the different name of LNP identified through the bibliographic research. And in the column, the different features we have identified, such as the size, the structure, or the main components of the nanoparticles. So as a result, we have had a three-phase approach through this abstract. First, we elaborated a database composed of 30 articles devoted to the characterization of lipid nanoparticles, as well as the methods employed to produce them. So a total of 39 features specific to LNP were extracted and gathered on a binary matrix. So for instance, the selected features were the size, the lipid structure, one layer, bilayer, lamellar or non-lamellar, and so on. Then the matrix was used to produce a dendrogram by Jacob index, a statistical analysis. So a dendrogram is a diagram frequently used to illustrate the arrangement of groups or clusters generated by hierarchical grouping where the height represents the distance between the groups and the vertical axis indicate a distance or dissimilarity measure. So therefore, we can conclude that the smaller the link height, the more similar the two connected groups are. So as a result, in this case, the Dendrogram measured the similarity between LNP and the 39 features we have identified. So as you can see in the slide, the elaborated Dendrogram enables us to split up the different types of LNP into two main groups. First, the red one and then the green one, as you can see here. We can first use this tool to position specific LNP unusual in literature with the one which are the most similar and emphasized. So therefore, it can be used to highlight some similarity which do not come forward immediately between two LNPs. So the obtained Dendrogram was indeed in integration with the scientific committee. As a matter of fact, we can see that 4 types of solid lipid nanoparticles were on the same cluster, and on the same way here, we can see that the 4 sorts of liposome are also grouped together, thus demonstrating that our Dendrogram emphasizes the same results as it was highlighted in the literature. However, care should be taken that this tool is based on extracted information, so there could be some missing information that could lead to potential bias. Therefore, for a more relevant tool, there is a need for all data for a better classification. So to conclude, this statistical tool could be used to position a new type of lipids nanoparticle with respect to the most studied structure and emphasize a non-unusual similarity. So this tool can be used by all scientists to bring more reproducibility and understanding on specific terminology of lipid-based nanoparticle. But also some other statistical methods could be used, such as the multiple correspondence analysis, which can bring the same result, but in this abstract we have chosen to more focus on the Dendrogram approach. So if you have any question, please be free to email us with the email address you can see here in the slide, or if you want to discover what CYBERNano does, you can see here the website address, so do not hesitate if you have any question.
Hello again. Thank you, Jeanne, for this very clear presentation. Just need to check if we have already questions or not. Just because Thierry sent at first the abstract and you presented today, can you just present yourself and say what is your role within CYBERNano, please?
Yes, no problem. I work for CYBERNano for almost half and 1 year. I am a research engineer in nanobiology and quality by design. So yeah, Thierry has some courses today, so that's why he couldn't attend to it. So that's why I presented it today.
We are glad to have you, and we are glad to have CYBERNano also represented, because you're a very active member of the ETPN community and very active also in participating to several European projects, notably when it comes to quality by design. So a very trustable partner. Thank you for that.
Thank you for having me.
Okay, so I think we don't have question at this time. This is classical hypoglycemia before lunch. I get used to it. So we'll
Alexander, I sent you one. You have to check your chat because I can't put it in Q&A.
Okay, fair enough, Ruth. But as a panelist, the rule is that you can speak whenever you want. So this is the
okay. Now, my question is, could this easily be transferred to other classes of nanoparticles if you have the literature information for input?
Yeah, I think it could be applied to all kind of nanoparticle. You just have to select the features that you want and then use the statistical tool. So yeah, I think it could be used to any kind.
Cool.
Yeah.
Thanks a lot to both of you, and thanks again, Jeanne.
Thank you.
So now we'll move to the next one. If he can hear me, is Jeanne Olaetxea from CYBERNano in Spain. Are you here?
Hi.
Hello. And yeah, we can hear you and see you perfectly. And even if it's not a competition and if it's a silly detail, you have been ranked #1 among all the abstracts that we have received. So congrats for that. Yes.
Thank you very much.
Yeah. Yes, Andreas, yes. So congrats for that. And we are listening to your lecture, and I hope that we will have questions at the end. So your lecture is about photonic monitoring of perinatal asphyxia. And we'll back in a couple of minutes. Well, not a couple, in 7 minutes.
Thank the committee for accepting and selecting our project for this Pitch Me Up session. I'm Jeanne Olaetxea, PhD student in the nanoengineering group of Andreas Seifert at nanoGUNE in San Sebastian in Spain. And in the following presentation, I will be talking about photonic monitoring of perinatal asphyxia. So in this project, we have a clear objective, which is not other than reducing the global neonatal morbidity and mortality rate, as well as reducing the complications that may derive from perinatal asphyxia. So based on this vision, on this objective, we have defined our mission, which consists in developing a new disruptive diagnostic method that allows us to detect perinatal asphyxia by continuous, non-invasive, and real-time monitoring of the baby. So let me introduce you the central problem. Perinatal asphyxia is one of the most common medical disorders in high-risk births, and it's characterized by an insufficient oxygen supply to fetal tissues. The problem is that if this episode is prolonged in time, perinatal asphyxia is responsible for neurological damage, better known as hypoxic ischemic encephalopathy. In order you to get an idea of how dangerous this medical disorder can be, in 2017, out of the 2.5 million neonatal deaths, 24% were due to perinatal asphyxia. And from those babies who survived, 1.5 per 1,000 live births suffered from this type of encephalopathy. So as you may have noticed, fetal monitoring plays a major role in the prevention and the diagnosis of perinatal asphyxia. However, the performance of the current standard methods, such as cardiotocography or fetal scalp blood sampling, which is based on pH and lactate analysis, they are far from being ideal, to such an extent that their use in obstetric care has been questioned. Why? Because they present a number of disadvantages. Mainly, we can say that it's a discontinuous, an intermittent, and an invasive method, that it often leads to a high failure rate in the diagnosis of perinatal asphyxia. Therefore, there is a lack of evidence on the effectiveness of these methods. Moreover, the surgical response that the obstetrician performs in order to avoid the consequences of perinatal asphyxia is one of the reasons, one of the causes in the increment in cesarean section and operative deliveries. The problem is that although the World Health Organization claims that the number of cesareans should not exceed 15%, the average rate of cesareans worldwide is above 22%. But where does this project come from? Why did we decide to address this social problem? Well, the project arises from a real problem of an obstetrician in his daily work, where he realized that there was a deficiency in the current methods for fetal monitoring. Therefore, he decided to contact us, and this is how a collaboration started between us, between the nanoengineering group, Biodonostia, and the Hospital University of Donosti. In this multidisciplinary team, the nanoengineering group contributes with expertise and experience in data analysis and in the field of optics and photonics. And this is how our solution, our technology is born. It consists on the combination of a Raman spectroscopy system with an integrated Raman probe and a computer or software based on machine learning algorithms. In order to protect our solution, to protect our technology, we recently filed a patent application. But why should we choose our solution? How does our solution differ from current methods? Well, easy answer, right? Our method, our diagnostic method, offers a continuous, a non-invasive, and real-time monitoring of the baby. And of course, this has a clear social impact in the society. How? By providing a more reliable and higher performance diagnosis of perinatal asphyxia that reduces the risk during the delivery process. Of course, this is done with a reusable Raman probe. If we have a look at the mid and long-term objectives, our technology expects to reduce the neonatal and maternal morbidity and mortality rate that implicitly reduce all legal, social, and administrative implications that are associated with neonatal adverse outcomes caused by perinatal asphyxia. Finally, we also expect that our technology reduces the personal and public expenditure intended to cover all expenses caused by such a situation. The chances of our technology to grow are clear and evident. As I said, our technology is satisfying a clear market need, and the market of medical devices is a market which is very receptive to technological innovation. Moreover, its size is huge. To get an idea, in 2019, the market of medical devices generated $457 billion. And by 2023, the expectations are that this number exceeds 600 billion. Regarding the proof of concept, for us it's very important to fulfill all the validation steps. For the hardware design and development, we have validated different Raman customized systems, and we have also validated our first Raman probe prototype. We have also validated our technology in vitro and ex vivo, where we were able to detect pH and lactate variations in body fluids and in aqueous solutions. Right now, we are currently working in the in vivo validation process with animals, and we expect to start with validation in humans the next year. We have also performed a retrospective data analysis where we studied the contribution, the correlation of different biochemical parameters in the diagnosis of perinatal asphyxia, since we really believe that data analysis performs a key role in the success of this project. So where do we go? Well, as I said, we are involved in the validation process that we expect that it will last 2 years. For that, we are looking for investors for a seed funding that we estimated at 900K euros. From these investors, apart from the capital investment, we also expect to get some support to elaborate a strategic plan that will help us to define our priorities and objectives. Of course, we also hope that they will help us to define the roadmap for the creation of a startup. I would like to end by saying thank you to our collaborators from Biodonostia, saying thank you to Eusko Jaurlaritza for the funding, and of course, thanking my team in the Nano Engineering Group. Thank you very much for listening.
Well, Jeanne, very, very, very good talk. Thank you for that. It's quite clear why you were so well ranked. I'm just checking if we already have some questions right now or not. Yes, someone is raising the hand. I think it's Joseph Coste. So Professor Coste, if you have a question, feel free to ask.
Yes, thank you. My question would be, I guess you are analyzing the amniotic fluid. What is the analyte that you are really looking at? What are the analytes for the Raman signal?
We are actually, the measurement that we perform, we don't look at the amniotic fluid, but we place our probe in the scalp, in the head of the baby, and we look at the systemic picture of the physiology of the baby. So instead of looking at just one parameter, our objective is to look at the all the changes that are happening in the physiology of the baby.
So how can you access the scalp of the baby?
I mean, this.
That's
yeah?
after birth or before?
I
was thinking this is something which is preborn baby.
It's just at the moment of the delivery. I mean, it can last a few hours. So at that moment, instead of performing the current methods, that they just make an incision in the scalp of the baby, which is accessible. Instead of doing this incision, they place the probe. So we have access to the scalp of the baby.
Thank you.
Yeah, you are welcome.
I do have a question.
Mm-hmm.
I mean, the medical need is extremely clear. Your technology seems to be really to show some unique features indeed. I mean, to date, what is the main block road for you? What is the main problem? Is it just about clinical development? Or, I mean, do you see other kind of problem when you try to create this startup company? And what kind of support are you looking for? What kind of help?
I mean, for now, as I said, we are validating our system in animals. So I guess in real cases we will have more problems because it will not be that easy to the conditions in the moment of the delivery are quite difficult. So I guess to get a real signal in vivo in those real cases, it will be much more difficult. And regarding you said what we are looking for in terms of investment, or what was your question?
In terms of support, I mean, do you lack partners for the clinical development? Do you need more money right now? What is the key things that you are looking for, and you might have some problem to find?
I mean, for now, we have the money that we received was from public funding, and what we are looking for is for investors that support this creation of a startup company.
Okay, thank you so much. Am I right when I said that you are still a PhD student?
Yeah, you are right.
Well,
you're right.
Looks like a very nice PhD thesis. So we wish you very good luck for the end of it and for the project that is really exciting. I mean, the medical need is here and technology seems to be very good. So thank you again for this great presentation.
Okay, thank you to you. Bye.
Bye. Well, stay here, stay around.
Yeah, I will be here.
And now for the last pitch, I will call on the virtual stage Jason Thomas Duskey from University of Modena, best place to eat in the world. Can you come and open your mic and video, please, Jason?
Good morning.
Good morning. So that's great that we have here Jason live, because you accepted a bit last minute to give a talk because one of the speakers could not make it today. So you accepted it. Thank you. Thanks a lot for the great efforts. And so you have the privilege not to do a pre-recorded lecture, but a live lecture. So we take maximum risk for the last one, okay? Like we are crazy people, right? So you can share your screen right now and we are listening to you. Please take the time.
Okay. So good morning, everyone. Thanks for having me, even if it was a last-minute invite. Thanks for sticking around for the final presentation, and thanks to the ETPN committee for allowing me to talk today about our lab's attempts to use the knowledge that we've already accumulated for nanomedicine in order to move into GBM treatments and to try to expand our options for what we can do against glioblastoma and other brain tumors. So as it was just introduced, I work at the University of Modena. We work in a type of consortium between 70 principal investigators, more than 40 labs, and around 280 people in order to produce new nanobiomaterials, diagnostics, and therapeutics. In general, we look at difficult to treat diseases such as brain diseases, and we try to link not only the target and the drugs needed to treat them but also the early stage sensing, detection, diagnosis, and imaging. For this project, we're talking about glioblastoma multiforme, and we're trying to improve delivery, specificity towards the cancerous cells over healthy tissues. But as you'll see in the later slides, we're very much looking at the tunability of our systems, not to be just one-shot cure if it arrives to that, but more to be very flexible in what we have to do in order to treat different types and different stages of the diseases, as well as early diagnostics. Some of the difficulties in treating diseases like these, like GBM, is that we have to cross the BBB, which, as everybody here knows, is difficult because we have the efflux transporters, we have tight junctions, and we have to cross. But it becomes even more complicated because in GBM, depending on the stage of the disease, you can either have a normal blood-brain barrier, a damaged blood-brain barrier, or a blood-brain barrier that's basically destroyed. And for each stage of the disease, we have to look at treating it differently because we have different barriers to overcome. Current solutions and patient prospects are pretty dim, even though the first treatments for this diseases were found more than 50 years ago, around the 70s. They have very limited effects. 5 in 100,000 people will be diagnosed with this, and one of the major problems is it's usually diagnosed at a very late stage, meaning that most patients will live maybe a year, 12 to 15 months, with less than 3% surviving for up to 3 years. And as I said, the drugs available have minimum effects and can extend life expectancy somewhat, but for a cure, we're very, very much limited in our prospects. And it also comes to say that during these short times after diagnosis, a huge portion of the cost becomes paying for the facility fees and the hospital fees. So it's very useful for us if we can start early diagnostics and have better treatments in order to not only save the patients, but also save the families and those involved the costs of treating the disease. Our solution is a little bit different than normal because we're not looking at just jumping in and forming a one-shot cure, but we're looking more at a complete system or a tunable system based on flexibility and translatability. So we're looking at improving targeting in order to get to the brain, as well as target the GBM cells themselves. But we're also looking at stepping back to the point of formulation and looking at improved drug compatibility, as well as nanoformulation improvements, which will allow us to not only have one system that works for one disease or one disease state, but a much more expandable system that we can rapidly change in order to be effective for many different types or stages of the disease. This is where it becomes unique, is instead of just looking for the cure and the physiological effect, we're more looking at the formulation aspect and the very basic of the nanomedicine formation. Proof of principle wise, we just started looking at GBM and treatments of GBM. However, as I said, since we're looking at the actual formulation based of the system, we have a lot of experience. We've already shown that with certain targeting ligands, shown here in the bottom left, we can cross the BBB in MPS 1 and 2 mice, as well as healthy animals. This has been shown not only to pass with small molecule fluorophores or drugs, but also up to the size of large proteins and enzymes. We've recently synthesized 2 other peptides that have been shown to cross the BBB using the opioid receptor and a non-saturable method. And this has allowed us to move on to like different drugs and trying to dose different drugs, such as peptides against Alzheimer's disease, as well as other small molecule drugs. However, we still have to work on the nanoformulations, because when we move on to things such as DNA, large enzymes, or incompatible drugs due to solubility, we have very much difficult in encapsulation or delivery. For this, we also look at producing hybrid nanoparticles or compounds that are conjugated to have different effects. Recently, we conjugated PLGA with chitosan in order to be able to bind DNA and possibly transport DNA also to the brain. And looking at the formulation method itself, we see that things such as enzymes or certain drugs, small molecules, are very susceptible to degradation during formulation process or during dosing. And by optimizing the formulation process and minimizing these effects, we've been able to see a direct link in dosing enzymes and having a physiological effect in order to reduce GAGs in the brain, and to combine all of these together to make a system that is very versatile. So next steps is we want to take these systems that we've already produced and move towards clinical trials, and therefore we can kind of get a sense of what we're missing or what is still lacking. And obviously the translatability of this will be the ability to add GBM, anti-GBM molecules or GBM targeting ligands that we have recently formed collaborations with in order to high throughput screen new ligands and new anti-GBM molecules in order to use this information that we've already produced to continue to expand our possibilities to treat GBM at different stages or have early detection systems. This will allow us to maximize all the parts from the drug to the pathology to the different production as well as scale up, and hopefully lead to more translatable clinical applications for a variety of different diseases or disease states. With that, I'd like to thank my lab and all the funding and collaboration groups that we work with in order to produce all of these results, and all of you for being here and accepting my talk today so that I could share this information with you.
Thank you very much, Jason. That was really nice. Except the last slide with the logo of the 60s from the ETPN. I will send you the new one. No, that was just a joke. Thank you for quoting us. That's a pleasure to be able to help our members. Do we have any question at this stage for Jason? You can open your video if you wish. I think we do. I think we do, yes. Okay, so Rosario is asking a question. 3 questions for the same question, 4 of them. Okay, I will try to read it. So, very interesting project. Could you please comment about preclinical related to toxicity and immune response?
So the final results we haven't gotten yet. Those are pretty young projects in the preclinical sense. However, as a part of the nanoformulation process and testing all the different types of particles that we make and hybrids and the effects, we test them in vitro and we look at whether or not certain hybrids or certain formulations cause immune response. And we try to minimize that depending on which disease state we're looking at. The ones that we have in preclinical trials now, we've shown that have very, very minimal immune responses, or they activate the immune system in very, very low quantities, if at all. And that's kind of one of the things that we try with every new system and expanding all of our repertoire to say is that we don't have to worry about this, because we know that if we have a drug that causes an immune response, we can create a nanoparticle that can contain that or minimize the effect. And we see this not only through the overexpression of immune cells, but also we can sort of control this, or we're looking at the effects of being able to control which proteins bind these nanoparticles in the bloodstream to create the protein corona, which has been shown to change how the immune response affects to these different types of particles.
Excellent. And we have a second question for you from Claudia Martins. Congratulations for the excellent project. Always cool. Would you give preference to a single targeting ligand that targets both the BBM and the GBM, or a dual targeting ligand system in which a first ligand may target the BBB firstly, and a second ligand may further target the GBM secondly?
That's a great question, because we've asked ourselves that as well. Which would I prefer? Neither. If we have a system like this, probably the goal would almost be to use both. So we're looking at multifunctional systems already. If we have also, we're looking at systems where it's not just 2 ligands, but it could be even more. Using different ligands that cross the BBB in together in order to use different uptake or BBB crossing methods, whether it be receptor mediated or clathrin mediated, different methods, and then also adding the GBM specific. It's hard to say because there's not really a preference. If one works better than the other, obviously we're going to go with the one that gives us the better results. But we're not limited to say one or the other, as we have the capacity to do pre or post modification of these nanoparticles with ligands, affording us the ability to continue to add more than even 1 or 2 or 3 antibodies or peptide ligands together in order to create a system that could potentially be even better than just one or the other.
Okay, great. Well, with this, I thank you again, Jason, for having accepted last minute. But it was really an amazing talk and a very interesting technology. Wish you the best for the development of this. And I would like first to thank all the speakers of this session. Before you all go to have lunch, we are going to close the normal part of the ETPN 2020 virtual event. On my side, before you go, I would like to thank again Spiros and Georgina, who are fantastic partners to organize events. But now we leave the floor to Ruchmit for some closing words.